Okay, folks, let's get started. Let's get started. How's everybody doing? Michael Gilson, are you here? I don't see you. Michael? I have an ID card that you might want. Michael, if you see this video, okay, we have your card, and it's being held for ransom, so I'll get something out of that maybe. Okay, let's see. Number one announcement. I will be hosting my occasional question session this afternoon. So if anybody wants to come and discuss something we discussed here or discuss the weather or whatever, that'll be this afternoon, 4 o'clock in ALS 2018. So if that's of interest to you, come there. I do videotape those, so if you really want to see what we're talking about, then you'll see it, assuming the videotape works and everything. Okay. We're moving along fairly rapidly, and there's a decent chance I'll actually finish early today, which we can either quit early, which I know you won't want to do, or we can start on new material. So we'll see how that goes as we get going along. No. All right. The next material is the last of the metabolic material, the last of the metabolism. So, yeah, so that might be good to just get in and get done, right? No. All right. Well, we'll see. We'll see how it goes. I do have a couple stories today, so that once I get long-winded in my stories, you never know what happens. All right. Well, last time I talked about the considerations in fatty acid biosynthesis, and I've talked to a couple of you now who have concerns about, well, there's not an awful lot of understanding of regulation of oxidation and synthesis and so forth. And to be honest, there's not. There's not an awful lot of regulation of them. Partly that's because they're fairly sequestered. Fats, of course, are stored in specialized fat cells called adipocytes. And they're not as readily available as are things like glucose and so forth. So that's one of the reasons that we see this happening. The other is that there are some controls, and one of the controls I've talked about with respect to fatty acid oxidation was the release of fatty acids from fats. That's controlled by the hormone-sensitive triacylglycerol lipase. And today I'm going to talk a little bit about the control of acetyl-CoA carboxylase, which is the only enzyme that is regulated in the synthesis of fat. Okay? So acetyl-CoA carboxylase, I'll remind you, is the enzyme that catalyzed that reaction that made that Malonyl-CoA. So the reaction that it catalyzed, and I'll show you here, the reaction that it catalyzed is this one right here. No, that's not the one, sorry. It's this one, okay? It's this reaction, acetyl-CoA going to Malonyl-CoA. Notice it's still CoA at that point, all right? So acetyl-CoA carboxylase catalyzes this reaction. One of the things that you heard in the song the other day, in fact, a couple things you heard in the song we hadn't talked about, and one of them is that this is like the pyruvate carboxylase that we talked about last term. This is an enzyme that adds a CO2 onto something, and so enzymes that add CO2s onto other groups almost always have a biotin coenzyme, and this is no exception. So this guy uses biotin to carry and attach the CO2 from this bicarbonate onto this acetyl-CoA to make a Malonyl-CoA. So biotin is the coenzyme, and virtually any time in a cell where you see the enzyme name is carboxylase, you may almost always assume that biotin is the coenzyme that's involved in that process. Now, acetyl-CoA carboxylase is a very interesting enzyme. As I say, and I know I say that about all the enzymes, but it's a very interesting enzyme also. And the reason that this enzyme is of interest partly is because, of course, it's determining whether or not you're making fatty acids, and also because of the controls that are on this enzyme. So what you see on the screen is a summary of the way in which the enzyme is inactivated going to the right and activated going to the left. Now we'll see it's actually a little bit more complicated than this, but this is a simple summary. You'll remember I said that the enzyme can be both covalently modified and allosterically regulated. So clearly what you're seeing on the screen are covalent modifications. Addition of a phosphate, removal of a phosphate. Okay? Now, I'd also like you to notice the name of the enzyme that puts the phosphate onto the carboxylase. Notice it is an AMP-activated protein kinase. That is not the same as a cyclic AMP-activated protein kinase. Okay? A cyclic AMP-activated protein kinase is going to be active when cyclic AMP is present. And when is cyclic AMP going to be present? When signaling has activated the adenylate cyclase, right? So this process is not driven by signaling. It's driven by something else. And something else is AMP. Now, remembering that AMP is not cyclic AMP, when do we have AMP present in cells? When there's an energy consideration, yeah? So when is, with respect to energy, when is AMP present in cells? When you have low energy, right? So when cells start running out of energy, they start converting ADP into ATP, and they do that making as a byproduct AMP. So when cells are low in energy, AMP concentrations will be high. And notice what's happening here. AMP is activating this enzyme, which is in fact putting a phosphate onto here and turning it off. This makes perfect sense. You don't want to be making fatty acids when the cell is starving to death, because making fatty acids takes a lot of energy, and the cell is trying to find energy, right? The cell wants to use all of the acetyl-CoA that it can in the citric acid cycle to make ATP, or make a proton gradient which makes ATP. Okay? So that's important consideration. Going the other direction, we see that protein phosphatase 2A, which is a phosphatase, that's not the same as the phosphoprotein phosphatase that we saw before. Protein phosphatase 2A is, takes the phosphate off, which converts it from the inactive to the active form. Okay? Now, this consideration will happen under what conditions in the cell? When energy is high. Okay? So obviously, when energy is high, we want the carboxylase to be active, or maybe we don't, depending upon our how much we have around here to begin with, like I have. We don't want this to be active, but maybe it becomes active so it saves fat for that rainy day. Okay? The problem is, like with me, I don't get enough rainy days, you know, and I live in Oregon, so I don't know. I need to. It's a bad. That was my joke for the day. Felt kind of flat, didn't it? Okay. Now, okay. So I've told you one of three things about the enzyme that I think is interesting. This isn't totally surprising. You've seen a regulation. You've seen regulation like this before. This one's driven by energy, pretty much. Okay. Driven pretty much by energy. Low energy turns it off. High energy turns it on. The second thing about this enzyme that's interesting is the fact that this enzyme exists in the cell as either monomeric units or as real long polymers. In fact, they're long enough polymers that you can actually see it in an electron microscope. Okay? So the individual subunits come together and make a long polymer. The long polymers are active. The monomeric subunits are not very active, and I'll show you what that means in a minute. Okay? So the long polymers are active. The monomeric subunits are not very active. That depends a bit on condition. Okay? Now, in order for the enzyme to polymerize, that is to make those long units, in order for the enzyme to polymerase, it's got to have the phosphate off. The phosphate's on, it will not polymerize. In fact, if you put the phosphate on, it will start coming apart because the phosphates repel each other and it becomes a monomeric unit. Okay? So there's two things about the enzyme. The third thing about the enzyme is its allosteric regulation. Okay? Now, on this schematic, you see that the molecule citrate can, in fact, partially activate the carboxylase. If there's a phosphate on there and there's nothing else present. Let's say there's no citrate present. If we're in this state over here, we have a completely inactive carboxylase. This is a monomeric unit. This is a monomeric unit, but it's a monomeric unit that has gained citrate, and citrate will activate that enzyme a little bit. Okay? Now, citrate, when are we going to have high citrate? Well, when the cell starts dumping citrate out of the mitochondrion because electron transport has stopped and oxidative phosphorylation has stopped because the cell is full of ATP. Right? So it makes sense that there would be, and remember that's the shuttle, the citrate shuttle is what brings acetyl-CoA out into the cytoplasm. It's no surprise that if we're dumping citrate into the cytoplasm, that citrate concentrations are going to be high, and we got to do something with that. We're going to take and make some fatty acids. All right? It's not going to be as active as if this phosphate were off, but it's going to be more active than this guy on the left is going to be. If the phosphate is off, citrate's going to help even more. Okay. Now, there's one other allosteric regulator of the enzyme, and the other allosteric regulator is a feedback inhibitor. Okay? What do you suppose the feedback inhibitor is of this enzyme? Okay, a specific one? Palmitate. Because palmitate is the end product of the fatty acid synthase. It's actually the most abundant fatty acid in the cell. So when the fatty acids start accumulating and they haven't been put onto fat, then it's time to stop this process because they're going to start making a mess inside the cell. Okay? So that's kind of cool. This shows the enzyme in 3 different forms. It shows it in a highly phosphorylated state. This shows it in an intermediate phosphorylated state and a dephosphorylated state. And we can see that in the dephosphorylated state it has the maximum activity, and we have varying levels of activity of the enzyme as we see here. Okay? Not surprising because this guy can polymerize. We can have many, many, many units, 1 or 2 phosphates. We have some, within those units we may have still some polymers. When we finally get down to more phosphorylated, we have virtually no polymers, and we're in the monomeric form. And the enzyme is very, relatively quite inactive. This is a log scale, by the way, so the activity is quite different between these. Yes, Carl?
Is there any specific reason why it's more active in the polymerized form?
Is there any specific reason why it's more active in the polymerized form? I guess the answer is because it is. There's... In the polymeric form, as you can imagine, you're going to have some slight changes in structure, and those changes in structure can, for example, allow access to the active site. Without those changes in structure, substrates will have difficulty getting to that active site. So the polymer, making the polymer actually favors that access. So that's the more technical answer to your question. Okay, clear as mud? Quiet group today. Okay, here's what some of these filaments look like. This is actually taken from an electron microscope, and you can actually see these things, okay? Those are long polymers of acetyl-CoA carboxylase. Pretty cool stuff. All right. Now, another thing you heard in the song yesterday is the fact that desaturation happens. And you saw when we had unsaturated fatty acids, and by the way, I think I said in the exam, I used desaturated and unsaturated in the same context. Desaturation being the way that we make these fatty acids with double bonds. Desaturation happens as a result of action of enzymes that are in the endoplasmic reticulum, and they're known as desaturases. Very simple name to remember. Desaturases are enzymes that are taking hydrogens and electrons from the single bonds in there. So, not surprisingly, they're going to have actually a very important component, FAD, being right here. FAD taking those electrons off of that molecule. And it's a complicated scheme that I'm not going to explain to you. It actually starts with NADH, but it results in the removal of electrons ultimately from over here to make FADH2. This scheme produces the very top product that you see here in one scheme, oleic acid. Okay, that's oleic acid linked to coenzyme A. Okay. There are other unsaturated fatty acids that can be made, but there are limits. And so I think I've mentioned in this class before that as mammals, we have limits on where we can put those unsaturated bonds in the fatty acid. The question on the exam about where, what limit did we have? We can only make double bonds beyond position 9. We can't make bonds beyond position 9. So we are stuck, therefore, with being able to make oleic acid, but we can't make other fatty acids that have double bonds further out, like the linoleic acid or linolenic acid. But we need these, fatty acids for the glycerol phospholipids and the sphingolipids in our membranes. So in order to have those, we have to have those in our diet, and it's for that reason that they're known as my mind's not working here today. Essential fatty acids. Okay, I knew it was in there somewhere. Okay, congested. Okay, maybe I'm catching this cold. I don't know. All right. So essential fatty acids have to be in our diet, and we need them because we can't make fatty acid. We can't make double bonds out beyond position number 9. All right. But the way that these things are made in all cells that can make them is through the use of desaturases, and the desaturases work by this rather complicated mechanism. Desaturases, as I said, work in the endoplasmic reticulum, and I have a question. Yes?
So we can't make double bonds beyond 9?
Yes?
How long is an unsaturated fatty acid? How long is there restriction?
Yeah, it's a good question. Can we make fatty acids that are longer than 16? And I haven't talked about that, and you actually give me an introduction to the next thing I'll be talking about. So let me say a word about that. We can make fatty acids that are longer than 16. So let me explain how that's done. Okay? All right. Also in the endoplasmic reticulum, we have enzymes that are known as elongases. Okay? So I don't have a figure for this. Elongases are nice and simple. And they simply add 2 carbons at a time to a growing fatty acid chain. They use Malonyl-CoA as well. And the mechanisms that they use are very, very similar to what you've already seen for fatty acid synthesis. The difference is that they're not part of that fatty acid synthase complex, because remember, that's in the cytoplasm. And the elongases are found in the endoplasmic reticulum. Okay? With elongases, we can make fatty acids that are considerably longer. We can make fatty acids typically up to as high as about 26 carbons. Okay? Pretty hard to find much bigger than that. But about 26 carbons, we can make fatty acids up to that length. Now, an interesting thing about the elongases is that they're found in the endoplasmic reticulum, but they're also found, surprisingly, in the mitochondria. Okay? Now, that's surprising because mitochondria is where beta oxidation is going on, and we worry about a vicious cycle, okay, or a futile cycle where we make and we break down, we make and we break down, and we make and we break down. But that doesn't happen in the mitochondria, and I've actually mentioned it earlier in class. That doesn't happen in the mitochondria because if you remember that the very first enzyme that's involved in fatty acid oxidation, that very first enzyme that only works on the long fatty acids, is found not in the mitochondria, but in the peroxisomes. Okay? So the acyl-CoA dehydrogenase, the long one, is not found in the mitochondria. So that means it's possible for the mitochondria to make longer fatty acids without going through a futile cycle of making them and breaking them down and making them and breaking them down. It simply makes them and they sit there, or they actually get moved out later, but they don't get broken down as a result of that. And this explains then partly why we see this division of where the acyl-CoA dehydrogenases are and why we have different ones that work on different fatty acids. Okay? Everybody got that? Pop quiz today? Yeses? You guys want a pop quiz? I was going to give you some extra credit. I guess I won't. So I wasn't going to give you a pub quiz. All right. Okay. Well, that finishes what I want to say about the fatty acids. I will point out, and they're listed down here, linolenate, linolate. I've already talked about these before. Palmitolate is another unsaturated fatty acid. It only has one double bond. And oleic, which is from oleic acid, the same thing right there. Okay. The thing that says study guide there, unfortunately, was the song we sang last time. I didn't get that off here this time, so we don't have a song to sing there, unfortunately. Okay. Well, talking about fatty acid metabolism, it's relevant. Oh, actually, I know what I wanted to say before I talk about this. Sorry, I just remembered. So one of the things I like to talk about this time is a common question I get from students, and that is, well, where do trans fats fit into the scheme? Okay? Where do trans fats fit in, and where do trans fatty acids fit into the scheme? And so when you hear the term trans fat, it means that that fat contains fatty acids that have trans double bonds in them, not cis double bonds. Okay? So you've got fats that contain trans fatty acids in them, not cis bonded fatty acids. Well, how do we get trans fatty acids? We don't. We don't make trans fatty acids. You saw trans bonds being made in the oxidation of fatty acids and the synthesis of fatty acids, but those were in fatty acids, not in fats. Remember, by the time we're done, we had saturated fatty acids, and when we desaturated them, we made cis bonds in every case. So where do the trans bonds come from? The trans bonds that you see come from chemical manipulation of the food by the food industry. Okay? Underline that. The chemical manipulation that makes trans fatty acids occurs as a byproduct of something called hydrogenation of vegetable oil. Okay? I challenge my students to go and look at any packaged food that you eat and look for the words partially hydrogenated blank. All right? You will be appalled at the number of places you find it, including in foods that say fat-free. Okay? Something that's fat-free doesn't mean it has no fat. It means it has below the level that they will call measurable fat. And even there you will find partially hydrogenated vegetable oil. Well, why do food companies partially hydrogenate vegetable oil? There are two reasons. One that they give is that it raises the melting point, so you can take oil and now have it be a little bit more solid at room temperature. Oleic acid, for example, is on the border of being solid or liquid at room temperature. Okay? If I take a polyunsaturated fatty acid, like linoleic or linolenic, and I remove a couple of those of the double bonds in there, it's more saturated than it was, and its melting point is going to be higher. Okay? So that may be important for, say, the texture of cookies that you're making from it, or the texture of some other food that you're making from it. And that was the original reason that people started doing this. Okay? Well, what they didn't realize when they were making these, they were desaturating these things, was that the hydrogenation was actually causing, as a byproduct, some of the bonds to flip into trans. Okay? So now a relatively minor component of that were trans fatty acids, and those trans fatty acids weren't free fatty acids, they were in the fats. So that's hence the term trans fat. Okay? Now, the second reason this is done, the food industry doesn't tell you. Okay? At least they don't jump up and down and say it very loudly. The reason that they don't tell you is because it keeps food lasting longer. Okay? Why does it keep food lasting longer? Well, because the little nasties that would normally get in there and eat your food don't like it. And they don't like it, but you are being fed this stuff. Okay? That kind of bothers me. All right? There's a lot of things that you think about that the little nasties may not want, and here you are eating it. All right? It makes it last longer. It keeps it from going bad. Okay? Yuck. That's kind of disgusting, isn't it? All right? Now, the more disgusting part of it is that trans fats in recent years have been implicated in some terrible things. Absolutely terrible things. Last term I railed about high fructose corn syrup. This term I'm going to rail about trans fats, because in the scheme of health, trans fats are probably worse for you than high fructose corn syrup, and you know what I think of that. Okay? Trans fats are linked to problems associated with cardiovascular disease. The more junk food that you eat, the more likely you are going to be aggravating that. They're implicated in inflammation in the arteries, which leads to cardiovascular disease. They're linked to increasing LDLs, decreasing HDLs, thereby taking away many benefits you might get from exercising. Okay? They're very nasty things. Now, I urge each of you to go look at your packaged food and look for the words partially hydrogenated blank. Okay? And then think about how much you want to eat of that stuff. Okay? Nasty stuff. All right, question, yes? Yeah, what happens that makes it so bad in the body? That's something that's an area of active research. I'm going to post on the website tonight a research, not a research article, but a research story from last year suggesting one way in which this might occur. One thing that is known about this is that trans fats appear to inhibit the production of one type of prostaglandin. It's a molecule that I'm getting ready to talk about here in a little bit. And that one type of prostaglandin is actually implicated in the progression of atherosclerosis. So let me save that for just a second to talk about that. But it's not really fully understood yet why they are implicated. But there is a relation, there is a correlation between the consumption of trans fats in the diet and in the development of atherosclerosis. Okay, other questions? All right, so that leads me to talking about prostaglandins. And prostaglandins are a group of molecules that in recent years have become very much into the public eye for a variety of reasons. And their functions are, I don't want to say bizarre, but they're bizarrely diverse. Okay? They're bizarrely diverse. They are implicated in things like cardiovascular health. They're implicated in things like heart health. They're implicated in pain. They're implicated in certain types of muscular contraction and other things as well. All right? So there's a lot of different areas that these things touch on. What you see on the screen is the way that we make prostaglandins. So prostaglandins are a class of molecules. There are many different prostaglandins. So they have different names. Typically, they have a letter after their name. Here's PGH2. That's one of the most important prostaglandins. And there are other ones that I'll show you a figure in just a minute. Okay? So each one has its own specific function. And what I want to talk about first is not about their function, but how they're made. Okay? So, as you can see on the scheme, the central molecule making prostaglandins going down and making a related group of molecules going up, called leukotrienes, is arachidonic acid. Arachidonic acid, you may or you may not remember from when I first talked about fatty acids, is a fatty acid that contains 20 carbons. It has 4 double bonds. The fact that it has 20 carbons gives it, these group of molecules the name eicosanoids. E-I-C-O-S-A-N-O-I-D-S. Eicosanoids are molecules that contain 20 carbons, and they include the leukotrienes and the prostaglandins. Okay, so, well, the question is then, well, when do we make prostaglandins? And we make prostaglandins as a result of the release of arachidonic acid from cell membranes. What's arachidonic acid doing in cell membranes? Well, glycerophospholipids and sphingolipids contain them. Most commonly glycerophospholipids. Okay? So the glycerophospholipids that are in the lipid bilayer are sitting there with arachidonic acid attached to them. And when that arachidonic acid gets released under certain conditions, then it can go through and make these pathways. Now, one of the things that arachidonic acid is also involved in is inflammation, swelling, as I said, pain, okay, a variety of things there. So what I'll hopefully tell you in a second will help you to see something about this. So here's the phospholipids. These are glycerol phospholipids in our membranes, and sphingolipids to a lesser extent. They get released by action of an enzyme, at least from the glycerol phospholipids, known as PLA2, phospholipase A2. Yeah, that's an important name. And you can call it PLA2 if you want to. That's fine. PLA2 releases the arachidonic acid from the phospholipid, and then there's no regulation. It goes and does its thing. It goes up, it goes down. Okay? The enzyme that converts arachidonate, I'm going to follow the down for the moment. Well, the other way we can make this is if we have diisoglycerols that are in the membrane, how might we have diisoglycerols in the membrane? What's that?
When hormone-sensitive triglycerol lipase clips off.
That would be one case where triacylglycerol lipase has clipped off one. There's another one from last term. It's what I was thinking of. Anybody? From what? Phospholipase C pathway. Actually, you're exactly right. So the signaling pathway we talked about last term generated diisoglycerol, and you probably memorized at that time as DAG. Okay, and that's sitting there in the membrane. So that can also be a source. In this case, we have a different enzyme that can release one of those fatty acids from there. This enzyme isn't as important in the scheme of things because we don't have as many diisoglycerols in the membrane as we do phospholipids in the membrane. But PLA2 is one I'm concerned about. I'll say something about it in a second. Arachidonate can go to prostaglandin H2 by action of an enzyme that comes in a variety of names. I shouldn't say the most common, but a common name among biochemists is prostaglandin synthase. It converts this guy to this guy. Okay? Now, as I'll show you in a figure in a second, this involves putting a bond to an oxygen in a ring. It involves putting a bond to an oxygen in a ring, and as a consequence, this enzyme, or this class of enzymes, is much more commonly known as cyclooxygenases. Cyclooxygenases. Now, cyclooxygenases get their name abbreviated, and they're called Cox enzymes, COX. So you may have heard of things called Cox inhibitors. Okay? Cox inhibitors Cox inhibitors are designed to inhibit this enzyme, stop the production of prostaglandin H2, and stop the production of a wide variety of prostaglandins. Cox inhibitors include things like aspirin. Aspirin inhibits cyclooxygenases, and since prostaglandins are implicated in pain formation, aspirin is a painkiller. It's referred to as a non-steroidal anti-inflammatory drug, and I'll give you an abbreviation for that. But if you want the whole thing, it's non-steroidal anti-inflammatory drug. And it's more commonly called an NSAID, NSAID. So NSAIDs are molecules that inhibit cyclooxygenases. Okay? They inhibit cyclooxygenases. NSAIDs work right here. Where you say, okay, these are the non-steroidal anti-inflammatory drugs. That must suggest that there are, in fact, steroidal anti-inflammatory drugs, and there are. And they're used more commonly for treating inflammation, okay, rather than aspirin, and they work right here. So steroids, you're taking cortisone or something like that, they're inhibiting this enzyme right here. So they prevent arachidonate from even being formed at all. I had poison oak. One of the first things I did when I came to Oregon was I went out picking blackberries in my shorts, and I didn't know what poison oak looked like. Okay, and I was miserable. I got poison oak. My leg literally doubled in size with the swelling from the poison oak. I was miserable, and they put me on cortisol so that I would inhibit this enzyme. because they were concerned it was going to get infected and I'd have to have my leg amputated. So I was very happy we had drugs that inhibited this. Okay. Once I started this stuff, it worked pretty quickly. Actually, the inflammation went down, a lot of pain went down, and so forth, and I was fine. I learned to look out for poison oak. Okay. Now, these enzymes, these Cox enzymes, are very interesting enzymes. Okay. This is a very simple picture of prostaglandin synthesis. Okay. When we look at prostaglandin synthesis, people have discovered over the years that there's really two main types of Cox enzymes. So one Cox enzyme is found pretty much in all the cells of your body. It's found in your stomach. Okay? And it turns out that one of the prostaglandins that your stomach makes is necessary to help stomach cells to proliferate. And stomach cells are some of the most rapidly proliferating cells in your body because they're constantly getting sloughed off by the very rough environment that they're in. So if you inhibit the production of prostaglandins in your stomach, over time, those cells are going to be replaced much more slowly, and you're going to favor damage, ulceration, and various wounds inside the stomach. So for some people, not all people, but for some people, taking aspirin over a long period of time causes stomach problems. This is why. So they're inhibiting the production of stomach prostaglandins that are necessary for the maintenance of stomach tissue. Okay? Well, people got looking around, and people who take aspirin over a long period of time don't take it, at least in big doses, because they've got a headache or they've got a backache. Well, maybe they take it for a backache, but they don't take it because they have a headache. They take it because they have chronic joint pain, rheumatoid arthritis, or some pain that is ongoing that aspirin or ibuprofen also fits into this scheme right here. Okay? Ibuprofen is also an NSAID. Over a long period of time, those cause problems in the stomach. Well, when they looked at the different Cox enzymes, it was then that they discovered there's this Cox enzyme called COX-2. So COX-1's found almost everywhere, including the stomach. They didn't find any COX-2 in the stomach. Guess where they found COX-2? In the joints. So all of a sudden, the drug companies went out and they spent billions of dollars looking for and manufacturing things called COX-2 inhibitors. And at first, it looked like they were absolutely magnificent. They were the wonder drug for pain relief, especially if you had rheumatoid arthritis. Because what you could do is you could inhibit the COX-2 enzymes that were probably associated with causing the pain. And by the way, the aspirin works on both COX-1 and COX-2 enzymes. Okay? But the COX-2 inhibitors were not inhibiting the COX-1 enzymes. So you're going to leave stomach alone. They looked perfect. Okay, there were several of them that came out. Celebrex, Vioxx. Okay, they hit the market 4 or 5 years ago with a splash. Okay, and it looked like that was the answer to the problem of the stomach issues here. Okay, and then they found at low levels people started having heart problems, valve problems. Okay, not a good career move. All right. And so most of them, not all of them, got taken off the market. For some people who have chronic pain, I think Celebrex is still out there. It can be prescribed and used in some cases. Vioxx got taken off. The drug companies which had spent billions of dollars invested in making these drugs basically had taken the shorts. They probably knew a little bit about it before they put them out there, so they weren't totally innocent. Okay? But nonetheless, they lost a major source of painkiller, this major source of revenue for them because of this involvement of prostaglandins in the heart tissue that wasn't known when they developed the drugs. Okay. Now, since then, there's been actually reports of another Cox enzyme called COX-3 enzymes, and I'm not sure what the distribution of those enzymes are. So there may be 3 classes of Cox enzymes. But anyway, that's the Cox enzyme report, cyclooxygenase.
So are you saying that the COX-2 enzyme was also in the heart, or that it sort of...
COX-2 enzyme, what it was just doing was inhibiting a heart enzyme that wasn't known previously.
Okay.
Yeah, and it was a COX-2 enzyme, yes. Okay? Yes, Jenny?
So aspirin inhibits both COX-1 and COX-2. Why doesn't it also lead to these heart issues?
It's a good question. It's a complicated answer, and it's probably a dosage effect. So the COX-2 inhibitors probably are much more effective at the dosages they were being used, and we're hitting that enzyme preferentially. But again, these are classes of enzymes, and there's many variants on them, so it's not completely clear why. Aspirin, I think I mentioned last term, aspirin is referred to as a blood thinner. And one of the reasons we refer to it as a blood thinner, it's actually taken by heart patients. And it's taken by heart patients because of a property I'll describe to you in just a second. But it's taken by heart patients to reduce the incidence of clotting and reduce other problems associated with heart attacks and things like that. The reason that aspirin is considered a blood thinner is that the prostaglandins can lead to a class of molecules called thromboxanes and other prostaglandins. Both of these are involved in making blood platelets sticky. The stickiness of blood platelets actually is a factor in the clotting process. So if you inhibit the production of prostaglandins that are making them sticky, they're less sticky, they're less likely to form random clots. Okay? And so it's for that reason that some people are told by their doctor, well, take like a half an aspirin a day to knock down the levels of prostaglandins and therefore reduce the likelihood you're going to have random clots. And that actually does some very, very good things for cardiovascular health. So these things are not damaging to the heart unless we're talking about, for some reason, the COX-2 inhibitors. Okay? Yes, Kaveen. That's what I just said. Yeah. So the aspirin is inhibiting the production of prostaglandins that are necessary for maintaining stomach tissue. Right? So people who take aspirin over a long period of time are knocking down prostaglandins that help keep stomach tissue healthy. And so when they don't have that, then the stomach tissue, it takes longer to be made. Okay? And therefore, you see less replacement, and you actually see ulcers and so forth that happen. Okay? Yes, Ryan.
How does aspirin help during a heart attack? Is that just to stop clotting?
During a heart attack, I can't say during a heart attack. I doubt there would be much effect during the heart attack. But again, anything that's reducing clotting is probably going to be a factor for that. The aspirin that's taken on a daily basis is taken over a longer period of time, so I doubt that it would have much immediate effects. Yes, Kyle.
Does most aspirin contain a little bit of acetaminophen and ibuprofen?
Does aspirin contain acetaminophen? In general, no. No. Acetaminophen actually works by a different mechanism, and it's not. Acetaminophen is one of the things that's not a Cox inhibitor. It works by a very unusual other mechanism. Acetaminophen actually has liver effects used in conjunction with alcohol. So you have to be careful with acetaminophen. But no, most aspirin does not have that. No. Okay, let's see. So as I said, thromboxanes are involved in making blood platelets sticky. I haven't said anything about the leukotrienes, and I want to say a word about those. The leukotrienes are also derived from arachidonic acid, and they are involved in a lot of things relating to allergies. So if you have, for example, asthma, as I do, some of the triggering molecules in asthma are actually leukotrienes. And these molecules can be present in very, very tiny concentration in the bloodstream and stimulate a very big inflammatory response in your nasal tissue, your lungs, etc. Okay? And so leukotrienes are implicated in that. One of the many strategies that's being tried right now for treating asthma, especially people who have very bad asthma, and mine's not very bad, but people who have very bad asthma, they're actually using leukotriene inhibitors that they've developed to stop an attack in the middle of an attack to keep people alive, literally. I actually knew an undergraduate student here at Oregon State who worked in one of our laboratories who died of an asthma attack. So it can be life-threatening. So you have to be aware of that. But anyway, the strategies for developing asthmas are focusing an awful lot on leukotrienes because of their involvement in asthma, among other things. So they can be involved in other inflammatory responses and immune responses that are necessary for cells. Okay, let's see. What else did I want to say there? Here are the eicosanoid structures that I promised you. And this shows us what the prostaglandins look like. So you say, well, that's an odd-looking molecule. Remember, this started out as a fatty acid that had 20 carbons, and it was linear. Okay? What the cyclooxygenases did was they created this ring, and they added oxygens. That's the the ring. Oxygenase, there's the oxygens there. And you can see that they have a variety of different forms. Okay, here's a very unusual. This is prostacyclin. There's leukotrienes. The leukotrienes are what I refer to as the linear pathway, because you'll notice the leukotrienes, remember they're going up, don't use cyclooxygenases and don't have a cyclic ring. Okay, they look sort of like prostaglandins, but they're not. The prostaglandins have this bond here that makes that a ring structure. Okay. And let's see. What else do I want to say there? Yeah. So the main thing I guess I want to say at this point is prostaglandins, the eicosanoids creating pain, are the ones to blame when you get inflamed and ouch, they hurt inside your brain. Prostaglandins, every throb and ache gets magnified. If you hope to win, cyclooxygens generation's got to be denied. The Vioxx has all been recalled, so go get yourself Tylenol. And if you ache, blame PGH synthase. We must complain that you make the aches. Prostaglandins, prostaglandin, D2F1G2, prostaglandin, it's you. Okay. Surprise.
Anti-leukotrienes.
Yes.
So if you're taking those, like something like Singular, does that have an impact on increasing prostaglandins or what? Because you have arachidonics to bring weight. So maybe like increasing
what I would say to that is you're making prostaglandins under specific conditions. You're not just constantly making prostaglandins.
Okay.
So one of the things I didn't say this year, which I usually talk about, are bee stings. So bee stings stimulate